So I welcome everyone to the My Tutor online school and to today's last lesson, I believe, which is in GCSE Biology. Well done for making it to Friday afternoon. The weekend is nearly here. So this session will be an hour long. It'll be about 45 minutes of content and 15 minutes of questions. And it'll be myself taking you through the whole thing. So I'm Charlotte. If you've been to some of the My Tutor online school biology lessons before, you'll have met me before, but if you haven't, welcome. So, a bit about me. I studied conservation and biodiversity at the University of Exeter. I've taught over 150 lessons on My Tutor and have been tutoring all of the biology sessions in the online school. So let's move on to what we're going to do today. So last time we had a lesson on Tuesday. We talked about stem cells and exam technique. And this time we're going to talk about microscopes and calculations. We're going to talk through converting between units and how to use standard form. And then that will be the last of the content from this module, which is Module 1, Cell Biology of the AQA exam specification. If you're not studying AQA or you don't know what you're studying yet because you're going to go into more GCSE biology next year, then don't worry. We've tried to keep the content quite general. It's generally stuff that is covered across all of the exam boards. So don't worry about that. Equally, if you're looking at taking GCSE from taking biology from GCSE to A-level, this is a really good reminder of all of that kind of basic stuff that you're going to be recovering again next year, but in a bit more detail. So if you look at the bottoms of your screens, you should have a chat and a Q&A box or available to click. So for that, any kind of little mini quizzes or things that we do, pop those in the chat. Any questions about the content themselves, put them in the Q&A. And I'll run through those at stages as we go along and make sure that I try and answer most questions. Okay, so units. So generally in biology, you are converting between units of distance or weight. Most of what we've talked about so far and what we're going to cover today is units of distance. So everything from kilometers all the way down to nanometers right at the bottom. And what I wanted to do today is to kind of talk you through how each of those increments of distance fit in together. And I've got some handy diagrams for you as well, which you're welcome to take off the screen, or you can find them by doing a quick Google search or going to BBC Bitesize as well. I think they have some on there. They're really handy to have kind of something that you can stick on the wall even above your desk and just remind you of how to convert between units, because you need to know some of the differences between, particularly for biology GCSE, you need to be able to convert between millimeters and micrometers. Usually are the ones that you use, and occasionally nanometers. So let's start at the top with kilometers. So a kilometer is, or a kilometer is 1,000 meters. 1 meter is 100 centimeters. And the clue here is in some of the prefixes to the words. So centi means 100, milli means 1,000. So quite often you can get an idea of conversions from the words themselves. So 1 meter is 100 centimeters. 1 centimeter is 10 millimeters. But that means that there are just froze for a second there. Okay, so 1 centimeter is 10 millimeters. 1 millimeter is 1,000 micrometers. So this is the next one down. A micrometer is that funny little mu sign. You use it quite often when you're talking about cells in biology. 1 micrometer is 1,000 nanometers. So you only really need to remember the 100 when it comes to centimeters. The rest of them are all increments of 1,000. They've put a handy sentence on the bottom here. So 1 micrometer is 1,000 nanometers. So a nanometer is 0.000000001 meters, or written in standard form as 1 times 10 to the minus 9. We're going to carry on and do standard form in a bit. So if you're a bit confused about what that means, don't worry, we'll run through that in a second. So those are kind of your basics. There's a really nice table here of converting between meters and all of the others. I quite like this table because it makes it quite easy. So if you go along the top, so if you want to go from meters to meters, you times by 1. If you want to go from centimeters to meters, you divide by 100. Millimeters to meters, you divide by 1,000. Micrometers to meters is 1 million. Nanometers to meters is 1,000 million. So notice there that apart from the centimeters, which are the awkward ones, you're adding on 3 zeros each time because you're always moving in those increments of 1,000. So that's how you use that table. If you have a quick Google, you can probably find it and a bit clearer than this one. This is a bit blurry. And I don't know why those 2 squares are highlighted. That must have been what they were using for that question. You see there, they're moving from micrometers to millimeters. So you need to divide by 1,000. If you're going from small to big, you need to divide. Going from big to small, you need to times. But what they've highlighted there is that, so when you're moving from micrometers to millimeters, so big, you divide by 1,000. And when you're moving from millimeters to micrometers, big to small, you times by 1,000. So you can see there's a bit of a mirror image going on that table down this diagonal line. Equally, if you work better going downwards, this is a nice diagram showing how you can work either up or down, converting between them. So a centimeter to a millimeter, times by 10, then times by 1,000 to get to micrometers, and times by 1,000 again to nanometers, and equally going the other way, dividing by those same increments. So if you prefer to look at it like that and draw you can do this in an exam. If you get a question, you're trying to convert them and you just can't remember because you're in a rush, draw one of these diagrams if you know it. That helps, just on the side somewhere. Okay, so converting to standard form. So standard form is the way that we write numbers that are really, really big and have lots and lots of noughts. It's a way of making them lots neater. So the essence of it is moving the decimal point until there's 1 digit to the left of the decimal point. And they've got kind of a handy diagram here about moving left and right, depending on whether your exponent goes up or down. The easiest way to do this is to look at an example, and then you'll kind of see how it fits in. So 156,000 in standard form. We move that decimal point. So we move it 1, 2, 3, 4, 5 places to the left. 5 places to the left, exponent goes up. 1.56 times 10 to the 5. Okay. So 10 to the 5 is our way of saying how many noughts you want to put on, how many times 10 you need to times that number by. Equally, if you're moving the other way, this one is a very small number. So we're moving the decimal point to the right. So across 1, 2, 3, 4, 5, 6 times. So our exponent, which is the small number will be negative this time. So it's 5.3 times 10 to the minus 6. So that's how quite often we write really big or really small numbers in biology. And you tend to only see them when you're doing calculations on cells, particularly in what we're covering today, which is microscopes. Be asked to measure things and might need to convert them to standard form. You've probably done this in maths. For those of you who haven't, you'll probably get a more detailed account in maths, so don't worry if it doesn't make sense straight away. This is generally kind of the only really mathsy bit of biology that you get. Okay, so microscopes are something that you have probably used in practical lessons or will use in practical lessons, depending on what stage you're at. There are two types of microscope that you need to know about. The first are the normal ones that you use in a lab, so a light microscope. You've probably all used them before. If you haven't, they're really cool. They were developed in the mid-17th century, and they use a beam of light to form an image. That's why they're called a light microscope. They're really handy, they're relatively cheap. You've probably got them in the labs at school. You can use them almost anywhere, and you can magnify almost anything. There's no kind of restrictions on that. They magnify up to 2,000 times, so you can see an image up to 2,000 times closer, and they have a resolving power of 200 nanometers. And resolving is the detail. So magnifying is how much closer you can seem to be. Resolving is how much detail you can see it in. And that's what they generally look like. The other type of microscope is an electron microscope. These were invented in the 1930s and are seriously cool. They use a beam of electrons to form an image, and they allow us to see in a lot greater detail. So there's two different types. There's the transmission electron microscope that allows you to see 2D images. And there's also a scanning electron microscope, and that allows you to see 3D images. Okay, they're very expensive. You generally only see them in universities or medical research centres where research is being done. They need special conditions. They're quite often in their own special room, and you're only allowed in with certain permissions. They're very, very big. This one that you see here is actually quite small. But they have huge advantages in that they allow us to examine subcellular structures, i.e. organelles. So when you look through a light microscope, you can often see cells. You can't generally see much more than that. So for example, if you wanted to look at ribosomes, you can't really see them in very much detail in light microscopes. Whereas electron microscopes have really, really good detail. So they magnify up to 2 million times, and they have a resolution of between 0.2 nanometers for a transmission electron microscope and 10 nanometers for scanning microscopes. And the reason for the difference there, so scanning doesn't have quite the same resolution as transmission because it's looking at 3D generally, rather than 2D images. So for transmission microscopes, you take a very, very thin sample of, say, a cell. You pop it between your slides in some special kind of liquids, special substances, and then you can view that specimen on a slide, much as you would do with a light microscope. You might have done that before. Whereas for scanning, you can view things in 3D, and they have to be kind of prepared properly and things. They're very cool. So as an example of that, here is a scanning electron microscope of, I think, a pollen granule, this is. You can see how detailed it is and how you can see, it almost looks like the moon, but spikier. So you can see all of those different kind of crevices and little pock marks all over there. And whereas on a microscope, this is what that looks like. So you can still see it's something, but you don't have that detail, that resolution that you get from an electron microscope. So electron microscopes have been pretty groundbreaking for lots of different types of research. Magnification and resolution. We've kind of covered this. So magnification is making things look bigger, whereas resolution is your ability to distinguish between two separate points. So how much detail is shown. Okay. So for calculating magnification, I don't know whether you use these triangles. I assume that you do for equations. So for this triangle, actual size, which is A, is image size, which is I, divided by magnification. So if you want to find the actual size of something that they've given you in an exam, you need to divide the image size by the magnification, and those will both be given to you in the question. Remember that actual size is usually in micrometers, whereas image size is in millimeters. So here you're converting between units comes into play. When you're working an equation, everything needs to be in the same units, so you need to convert first. So you need to convert your millimeters to micrometers by dividing by 1,000, according to those tables that we saw on the first page. Oh, it looks as though my slide has merged these together. What I was trying to get across here is actual size is image size divided by magnification. But equally, if you're given any two parts of these triangle, you can find the third. So magnification on this side would be image size over actual size, so divided by actual size. And the image size would be actual size times the magnification. Okay. So that's those triangles. I find them really handy to remember. This one has quite a good acronym. You can call it the AIM triangle. Okay, got a couple of past questions to show you how that will work. I'll do a quick run through the Q&A, just see if anyone's got some immediate questions before we go on and do some practice questions on that. Okay, a couple of people saying, can I go back to slides? So let's put the light microscope slide on. We need microscopes for viruses and for vaccines. You can view viruses using electron microscopes. Okay. I'm saying that they can't hear me. If you're having trouble hearing me, it's probably an internet problem. My internet is fine. I have had a look at this. So see what you can do, either moving closer to the router, you might need to log back in again and see if that helps. And as I said at the beginning, this is AQA exam board, but it's generally common across all exam boards. Microscopes and cell biology are the first thing you learn in all of them. Okay, and I'm going to pop to the magnification definition at the top there. Okay, so let's look at some past exam questions. So, here's our first one. So as always, with exam questions, the first thing that we do is underline. So here we have the cheek cell in figure 2, which down here is magnified 250 times. The width of the cell is shown by the line D to E. Calculate the width of the cheek cell in micrometers. It's given you the units. And it's broken it down for you. This is quite a kind question. So it says complete the following steps. Measure the width of the cell using a ruler. Obviously, we can't do that today because we're all looking at computers. But I'll do the answers in the next page, and I've already measured it. And then it's given you the equation as well. So use the equation to work out the real width, so the actual size of the cell in millimeters. So real size is image size over magnification. And again, it's done millimeters. Then it's asked you to convert millimeters to micrometers. Notice that it's asked you to do this and it hasn't given you any indication of how, so you do need to know those conversions. So we've measured the width of the cell using a ruler and it was 45 millimeters, 4.5 centimeters. And then use the equation. So we've done real size was image size over magnification. So the image size was 45 millimeters over that magnification, which was 250. And that equals 0.18 millimeters. And then from 0.18, we needed to convert to micrometers. To do that, we needed to times by 1,000. So move that decimal point, 1, 2, 3 places, and you get 180 micrometers. That was 3 marks, that question. Quite a kind question. Nice, well done. Okay, can we keep the question and answer to questions about the content? Anything else in the chat? Okay. Next question. So let's underline this one again. A red blood cell is 8 micrometers in diameter. Diameter across. A bacterial cell is 40 times smaller. Calculate the diameter of the bacterial cell. And apologies for those of you that wanted to have a go first. I have put a red X in here. I thought I'd put it on the next slide. So the correct answer there is 0.2 micrometers. This one's kind of a bit different because it doesn't involve a kind of calculation using the using an equation. So you know that the bacterial cell is 40 times smaller, so 8 divided by 40 gives you 0.2 micrometers there. Again, 1 mark for that one, just a question of thinking it through. Okay. Next one here. I have got the answers on the next one this time, so I'll give it a pause and let you guys have a go, those of you that want to, just for a few seconds while I read it through, and then we'll move on to the next one, or the answers rather. So the image below shows some muscle cells from the walls of the stomach as seen through a light microscope. So you've got these funny little thin muscle cells with lots of mitochondria. They've given you a scale bar of 0.1 millimeters. Describe the function of muscle cells in the wall of the stomach. Should know this one. We have covered this in this topic. 2 marks for that. And then part B, the figure is highly magnified and the scale bar represents 0.1 millimeters. We'd already got that because we read the question properly. Use a ruler to measure the length of the scale bar and then calculate the magnification of the figure above. And I can tell you, I can't remember off the top of my head, I'm just going to flick. It's 40 millimeters. So this one was 40 millimeters. Okay, so if you want to have a quick go, I'll give you a few seconds. Calculate the magnification from those two numbers. They've been kind here, they've given it both to you in millimeters, so you don't need to do any converting of units. Good, someone's answered contract or shorten. It's a good answer to part A. I'll give you a few more seconds and then we'll reveal the answers. Good. Okay, let's have a look. Okay, so part A there. The function of muscle cells in the wall of the stomach is to contract repeatedly to mix food in the stomach. So you'd have got one mark for saying that they contract and the other for saying why they contract. So they contract repeatedly in order to mix food in the stomach and help with digestion. So well done those that got that one right. The figure above is highly magnified. So this is the part B, this question. So for this, I've kind of broken it down into steps. So magnification is the size of the image divided by the actual size. The image, 40 millimeters was 0.1 millimeters of the actual size. The magnification is the same across the whole image, so you didn't need to worry about measuring the actual cells themselves. So magnification is 40 divided by 0.1, which is 400 times, which a lot of you got, so well done. Fab. Great. Okay, so that was kind of a whistle-stop tour of some of the maths that you're likely to come across. So for the next bit, I have prepared a quiz for those of you that have been asking. So I thought it would be a good idea to kind of do a bit of a revision quiz of the whole topic. So the cell biology topic comprises of the difference between plant and animal cells, between eukaryotic and prokaryotic cells, and specialized cells, so root hair cells, xylem and phloem, muscle cells, sperm cells, and nerve cells we talked about. It also looks at transport in cells, so diffusion, osmosis, and active transport. Cell division by mitosis we looked at as well, and cell differentiation and specialization from stem cells. So we looked at stem cells and how they can be used in medicine. So I have a, I think, 10-question quiz about this. So I'm going to use quizzes, which I think some of you have used. So here we go. Play live. 10 questions. So use any device to open. I'll pop this link in the chat as well, so you can paste it onto your computers. Want to? Oh, hold on, I didn't put that to everyone. So pop that link, join myquiz.com/pro, enter the game code. You can see a few of you in here already. Good. Quiz is pretty good. It allows you to do lots of different types of questions. So it works quite well. So far I've been using Zoom kind of mini quizzes that we've just run through the chat. This allows a bit more variety. So. Okay. The link is in the chat. Go over to the chat if you're struggling to find it. Someone's put in a name that's no good. Just removed you. You can come back in if you put a acceptable name. Don't know why that would have changed. I need to try something different. Oh, Charlotte Fan Club, you can win. Just kidding. Okay, any more? There's nearly 30 of us in here. There should be a few more people coming in. So if you're struggling, let me pop that one as well. Pop it in the chat again. Pop that link in. You can either use your phones or your computers that you're on or your laptops. Pop the link in, pop a name down and enter this game code. So it's 194162. I just pop that in the chat as well if you need that. Good, there's a few more people. Nice one. Okay. If you want to answer through the chat, then yeah, go for it. There's no reason why not. If you can't get it to work, yeah, just pop it in the chat. That's fine. No problem. We'll start in a second. And you'll be able to see them on the screen, so it should be fine. Just pop them in the chat. Okay. No worries. Right, let's start. So we've got team in quizzes, and then we've also got the team on chat as well. So let's start. I put timers on some of these as well, so we'll see how it works. Ah, you don't see them while you're on. Turn my sound down. Okay, let's run through these. So there's some people doing them quite quickly. We'll run through them for those of you that want to do them on the chat. So, number one, tick the organelle that is found in plant cells but not animal cells. So you have to pick one. The options are cell wall, cell membrane, mitochondria, or ribosome. Tick it if you need, pop it in the chat if you want to. Organelle that's found in plant cells but not animal cells. Cell wall, cell membrane, mitochondria, or ribosome. Okay. You want to put them in the chat, then go for it. Yeah, that's no problem. Okay. Let's move on to the checkbox. Question here, question 2. Tick 3 features of a prokaryotic cell that are not found in eukaryotes. So 3 features of a prokaryotic cell that are not found in eukaryotes. Actually, another thing is, if you are struggling to do this and you want to just write it down while I read it out, we'll run through the answers, so no problem if you want to do that as well. Just challenge yourself that way without putting it in the chat, that's fine. So those 3 features of a prokaryotic cell that aren't found in eukaryotes: flagella, nucleus, plasmids, cell wall, or single-strand DNA. 3 of those: flagella, nucleus, plasmids, cell wall, single-strand DNA. Okay, well done. It's incorrect here. 3 features of a prokaryotic cell that are not found in eukaryotes. Let's move on to question 3. So question 3 is a fill in the blank. Cells that are undifferentiated are called something. What do we call cells that are undifferentiated? Okay. Number 4, another fill in the blank. Cells that have differentiated in order to perform a specific function are described as, another word, just one word. Cells that have differentiated in order to perform a specific function are described as. What do we think? So they've already differentiated so that they can perform a specific function. Checkbox for question 5. Tick 2 ways that root hair cells are specialised for their function. Feel free to tell me if I'm going too quickly. I think we're probably all right. Most of you, I think, have got through these. So 2 ways that root hair cells are specialised for their function. Your choices are large nucleus, large surface area, lots of mitochondria to provide energy for active transport, lots of chloroplasts for photosynthesis, no cell wall for ease of transport. Tick 2 of those. 2 ways that root hair cells are specialised for their function. Leave that one on the screen for a sec. Okay. Question 6, what are 3 things that can speed up the rate of diffusion? So tick 3 things from this list. Your choices are large surface area, lots of sunlight, short distance, low temperature, or large concentration gradient. Tick 3 of those. Okay. Number 7 is multiple choice. If the solution outside a cell is described as hypotonic to the cell, which way does water move by osmosis? Your choices are into the cell or out of the cell. So which way does the water go when the solution outside a cell is hypotonic? I have included a sneaky diagram there to help you if you're unsure what hypotonic means. Number 8 is linked to that. So when water moves into an animal cell by osmosis down the concentration gradient, the animal cell is in danger of bursting. If you remember, there's a diagram of red blood cells bursting. What happens when water moves into a plant cell in this way? Got 3 choices. The plant cell shrivels, the plant cell becomes flaccid, or the plant cell becomes turgid. You need to pick one of those. What do plant cells do when water moves into them? Okay. Finally, last 2. So number 9 is another multiple choice. What or which is the longest stage of the cell cycle? So stage 1, which is replication, stage 2, which is mitosis, or stage 3, which is the final division. Which takes the most amount of time? Okay. And finally, another fill in the blank. Cells that divide by mitosis produce 2, and I put XXX, which means 3 words. 3 words. There's a certain phrase that we use when we're describing the product of mitosis. Two things described by 3 words. Okay, well done everyone. 46% accuracy, good. Yes, well done for now. Identical daughter cells is exactly what we're looking at for question 10. Okay, let's have a look at the leaderboard. Well done, Jess. See yourselves on there. A couple of people still finishing up. Terrible names you've chosen. Keep going. You're still going. It's no problem. Plenty of time. Okay. Done. A few more people getting them right. Okay. 10 more seconds. Well done, everyone. A couple of people still moving through. We're going to call on into it in a second, and then we'll run through the answers. All right, we're going to have to call it there, I'm afraid. So we've got plenty of time to run through any areas where we didn't do so well. So I am going to end the game. There's a couple of you that haven't quite finished yet. Oh, so we're making rapid progress there. Okay, let's finish there. Okay, well done, you guys. Okay. So quite a few people struggling with question 2. 10. Can I do the answers now? Yes. Okay. Take the organelles found in plant cells but not animal cells. So the answer there was the cell wall. Cell membrane, mitochondria, and ribosome are all found in all of them. Three features of a prokaryotic cell that are not found in eukaryotes. So this is a slightly trick question. See, a few people picked cell wall. So remember, cell walls are also found in plant cells. Plant cells are eukaryotic cells. So things that are found in prokaryotic cells only, so bacterial cells only, are flagella. They're those weird tails that they can have. Plasmids, which are those circles of DNA, and single strand DNA. If you're marking these because you wrote them down, then hopefully got these right as well. As we're going along, if there's stuff that you're not sure about, then I know, I'm sorry guys, I know some of you are still playing. But we have to finish the lesson at 4:15, so we do need to finish. If you have any questions about the content that you want me to clarify, pop them in the question and answer. So if you're not sure why a question doesn't make sense, pop that in the Q&A and we can go through it in a sec when we've been through everything. Okay, question 3 was a fill in the blank. Cells that are undifferentiated are called, wow, loads of people put loads of different stuff in here. Stem cells was right. Stem cells was correct. So there's quite a few different answers in here. Most people seem to get stem cells. That's what we needed. If they're undifferentiated, they're not specialized. Okay. If you have a question, pop it in the Q&A. Okay, question 4. Cells that have differentiated in order to perform a specific function are described as specialized cells. Very good. Whoever put specialized, you should have had that one. There's a few different, it should have picked that up, so I'm sorry about those. They are specialized cells. Well done. Those that got those. There's a few of you that didn't get those right and should have done. So for example, SB there, you should have had specialized. So I'm sorry about that. Question 5, tick 2 ways that root hair cells are specialized for their function. Root hair cells have a large surface area. That's the whole function of their root hair. They have lots of mitochondria to provide energy for active transport as well. So if you remember, root hair cells are what takes the stuff from the soil and brings it in, active transports it into the root hair cell to transport to the rest of the plant. They don't have a particularly large nucleus. They don't have lots of chloroplasts because they're under the ground, so they can't photosynthesize. And they do still have a cell wall to help them keep their shape under the ground. Okay. 3 things that can speed up the rate of diffusion. Large surface area, short diffusion distance, and a large concentration gradient. We talked about this a lot. You might be quite sick of this. You don't need lots of sunlight to increase the rate of diffusion. Sometimes that helps because sometimes the sunlight raises the temperature, but it doesn't do it directly. And a low temperature doesn't help either. So if temperature is raised slightly, then sometimes the molecules move a bit faster, because if you remember, diffusion is kind of the passive movement of particles that are always moving anyway. So heat increases that energy. Question 7. If the solution outside a cell is hypotonic to the cell, which way does water move by osmosis? I'm really glad that 13 of you got this right. This was a hard question. So if a solution outside a cell is hypotonic, it means it's very dilute. So there's lots of water outside the cell, whereas inside the cell, there's a few more things dissolved into the water. So there's a lower proportion of water. So the water moves from an area of high concentration to low concentration down the concentration gradient. So it moves from where there's lots of water to where there's less. So it moves into the cell. And then when water moves into an animal cell by osmosis down the concentration gradient, it's in danger of bursting. What happens when water moves into a plant cell in this way? Plant cell becomes turgid. Really well done who did that. That's the way that plant cells like to be. They have that big vacuole to help them hold a lot of water. That lowers the concentration of water in the cell. So they like to have water coming in. They like to be nice and plump. Longest stage of the cell cycle is the first stage of replication. So it takes the cell a long time to kind of grow, to replicate all of its organelles. So it's got 2 copies of everything and 2 copies of all of those chromosomes. Then in mitosis, the chromosomes line up and then they move into 2 groups and you have 2 nucleuses. And then finally, the cell membrane divides around those nucleuses. stage 3. Cells that divide by mitosis produce 2 identical daughter cells. Daughter cells you could have there as well, no problem there. Identical daughter cell, oh, you should have had that high, sorry. I can only think of so many combinations when I do this. So it might have been that you just missed the S of cells there. Daughter cell, again, you should have had that. Sorry about that. Okay, identical daughter cells, well done everyone that got those. That was brilliant. Okay, that was the end of those ones. Okay, hopefully that was helpful. Toughest question was question 2, so if you got past that one, well done. Okay, let's have a look. No questions. You all know all of it. Well done. Okay, in that case, I've got some Slido things to do. If we haven't got any questions, we'll just spend 5 minutes doing those. Okay, so the first one, I'm going to put a link in the chat again. You want to pop this into your computer, that link there, you can see. And that'll bring up Slido screen. And it'll bring up a question, which is, what are the key words that you remember about different types of cell? You can see that on your screens. So pop an answer to that question. Well done, turgid, good, plant cells. Any more? Or a few people can put some more keywords in there. Different types of cell. What kind of different types of cells are there? We just heard a lot about them in the quiz. Should be able to know these. Well done. Flaccid, hypertonic, isotonic, specialized, differentiated. Brilliant. Okay, any other types of cells? What about plant cells and animal cells? What kind of organelles do they have? Let's put those in. What are prokaryotic cells? Let's put those in as well. Any more? Red blood cells, well done. Hair cells, yes. Root hair cells. What other types of cell? Bacteria cells, yes, good. Pop some more in. Nerve cells, well done. White blood cells. Good. What kind of different organelles can you have in cells? Immune organelle, egg and sperm cells, yeah, good, they're really specialized cells. Nice. Good. Cytoplasms coming in there. Brain cells, yeah. Well done. Cytoplasms, brilliant. Mitochondria, yeah, well done. Talked about lots of root hair cells. Fab. Nice. Well done, everyone. That's a really good collection of keywords that we need to remember. Vacuole in there, yeah. Okay, let's stop that one. Down. Let's do cell division. Probably be the last one that we do. So for this one, got another link. So the second link there, pop that one into your browser. You should see a question again. We'll create another word cloud. What are the keywords that you remember about cell division? Good, that's a good start. Division, replication, identical, yes. Split, good, yeah. What about the different stages? So we've got replication, daughter cells, well done. Chromosomes, yeah, really good. Identical daughter cells, good. That's a really good phrase that we've kind of learned today. Mitosis, exactly. And the final split there. Cytokinesis, yeah, really good you put that one in. That's the posh name for the last stage, stage 3, when the cell finally splits. So it's membrane, and if it's a plant cell, the cell wall move, and it becomes those 2 separate daughter cells. Remember that those daughter cells are identical in their DNA, so they are could be described as clones. They are completely the same genetically. Sun cells, yeah, we did talk about that. I don't know why they're called daughter cells. It doesn't make any sense. Brilliant. Okay. Let's leave that interface. Yeah, there are loads of different stages of mitosis. So if you go on to A level, you will learn lots more about the different stages of mitosis. To get lots of different ones there, and interphase is one of those. Okay. Let's stop that one then. Finish that one there. Well done, everyone. That's worked really well. So I feel like we've got quite a good knowledge of all of those topics. And next week we're moving on. So we're looking at photosynthesis. We're looking at a bit of, I think it's just photosynthesis next week. So that'll be the main topic for that. So definitely tune in. I think we're on Monday and Friday next week, but check out the timetable and that will let you know. And don't think there's anything else that you need to know. If you are struggling, remember that my tutor does one-to-one tutor support as well. So you can have a look through the website and you can find a tutor that might suit you, and check that with your parents. And that's it, I think, until next week. Well done, everyone, for today. I think we've got a good grasp of everything that we've done so far. And hopefully see you on Monday. Have a lovely weekend. Bye, everyone.